Thanks very much, and thank you, David, for organizing this mind-shaking conference and for inviting me to talk about our work on epigenetic inheritance. And also thank you for bringing us to this brand new, I heard, building. It's a beautiful new place, and I understand that your department chair was very helpful in that sense. So, and she's here, Rachel Crosby. So thank you. We should all be thankful to David and Rachel. I'm going to, now you've seen this title since 1 hour or 1 hour and a half, so you want to know what's behind it. So I'm going to tell us about our work on epigenetic inheritance and on a new concept we are working on about the possible mechanism of epigenetic inheritance based on chromatin. So we are all influenced by the environment, whether diet, whether our life experiences, mostly extreme experiences like traumatic stress in childhood, but also cigarette, alcohol, endocrine disruptors in the environment, but also positive parameters or factors like sports, social environment, and literally every cell in our body, whether brain cell, liver cell, heart cells, can be influenced, including germ cells. Now, here, I'm not talking about an influence on the genetic code, because what you eat, being exposed to traumatic stress, is not going to induce any mutation or rearrangement of your genome or any change in the genetic code, but in the epigenome. Now, if we think about exposure, if, let's say, this is a man or a woman who are exposed, so that's the first generation. It's them who may display or manifest symptoms of exposure. Now, if their germ cells can also be affected, if the epigenome in these germ cells, in particular if the person is exposed during early life, during childhood, when germ cells are, it's mostly germ stem cells, which are not completely protected in gonads, and the changes persist, I mean, then we are talking about direct exposure of the second generation. And if a woman is expecting, her baby may have also cells in his body or her body affected and express symptoms. Now, the baby herself or itself has future germ cells. So if indeed exposure is chronic enough or strong enough, perhaps the germ cells of the baby himself or herself are going to be affected. And then we are talking about the third generation. So here it's fairly easy to understand. It's direct exposure. Now, how about following generations? Is there a possibility that any change in the epigenome could really stay? Not necessarily all changes induced during exposure, but some changes may persist and then be somewhat embedded into the epigenome of germ cells and be responsible for exposure-induced phenotypes across multiple generations. So is there evidence in nature about the inheritance of acquired traits? Yes, there is lots of evidence in C. elegans, in some cases up to the 14th generation, in exposure to temperature, but there is also evidence in rodents, some so in relation to trauma, to paternal obesity, exposure to valproic acid. I will show you some of the data about postnatal trauma, evidence in mice and rats. And here, in endocrine disruptors, the lab of Michael Skinner, since almost 25 years, has been working on that, and other labs also. And there is also evidence in human, which is correlative, of course, but there are epidemiological and large historical cohorts looking at the influence of diet during early life on the health and life expectancy of children and grandchildren, and a correlation between the lifestyle and diet in grandparents and health of children and grandchildren has been drawn. One of the latest studies has been done on smoking. Now, the concept is the following. Let's say there is exposure, and exposure will induce changes in the epigenome, whether DNA methylation, histone modifications, non-coding RNAs, which are part, there are molecules which can regulate genome activity. They are not per se epigenetic factors, but we can generally englobe them into the mechanisms which regulate genome activity. There may be other mechanisms that we are not aware of or that are not studied. Here, DNA methylation, histone modification, and non-coding RNA are the most studied because they can be quantified. They can be measured now with multi-omics. So these changes in the epigenome may happen in brain cells, neurons, in glial, in any other cell, and in germ cells. Here I'm showing seminiferous tubules of testis, where you see this tube. It's a very beautiful structure where you have the germ stem cells on the outside and spermatogenesis, which goes on, and maturation, which leads to the maturation of the germ cells until the sperm cell, which is in the lumen of the tube for release. So these cells can also be modified. You see here a sperm cell, even if chromatin is different in sperm because it's, most histones are replaced by protamines put to make it more compact. DNA methylation exists in sperm cells, histone, protamine modifications, and non-coding RNA. So now, if these changes persist and are passed to the following generation, we could imagine that the following offspring, the grand offspring, great and grand offspring, may manifest phenotypes. So now, the question is, what could be the potential mechanism underlying the persistence of these effects? And the hypothesis is that the life experiences induce durable changes in the genome, and here I'd like to put forward this novel hypothesis of chromatin being one of the substrates for these persistent changes. In immune biology, it's known that immune cells can keep immune memory by changes in chromatin structure. So this concept has not been studied yet fully in neurobiology, and not even less in reproductive biology. So here you have a scheme of the different, this is the pointer. This is the pointer here. Yes. Okay. It's not connected to your laptop. Ah, okay, fine. So here you have the different levels of organization of chromatin, with on the left chromosome territories, then different compartments. Compartment A, which is heterochromatin, and B, urochromatin.
Use the pointer of your laptop.
Yeah, just people online won't see the. No, I think they should see. Okay, and here on the far extreme you have so DNA methylation, histone modifications, and non-coding RNA, which may be around. But there is an intermediate stage here where, depending on some histone modifications, chromatin can form loops, for instance, in between enhancer and promoter. So if you have enhancer and promoter which are far away, if they have the proper histone modifications, they can come together, form a loop, and then genes in the loop or in the vicinity of these enhancer-promoter interacting sequences will be differentially regulated. And it's been suggested that these loops can be persistent. A very interesting thing here is that enhancers, when they are activated, can be transcribed. And there are enhancer RNA which can be produced bidirectionally, and this enhancer RNA can keep also message and can feed back this message to their own enhancer and to nearby regulatory sequences. So here we have asked the question of whether chromatin structure itself, so changes in chromatin accessibility, could be induced, persistent changes in chromatin accessibility could be induced by exposure in germ cells and other cells, and whether these changes could be persistent enough to then establish a state of prime state of chromatin, which would then influence gene transcription differently at a later time point. And here you have it illustrated here. So chromatin can go from from the open state to closed state bidirectionally, and upon exposure or a very salient factor, this would be tilted in one direction or the other, and this chromatin looping would somehow be maintained, probably by binding of different genomic regulators, transcription factors. The details of how loops are maintained in the context of exposure is not known, but there are different possibilities. Now, the questions I'd like to address today are whether chromatin in brain cells can be affected by early life stress, whether chromatin could also be altered in reproductive cells, and whether molecular changes in reproductive cells affect the offspring. So I start by brain cells because after all it's a neuro-oriented symposium, but here it's a mixture. Our research actually, even if we are neurobiologists by training, our research now is more reproductive biology than neurobiology, but we try to do this type of analysis in parallel in brain cells and in reproductive cells. So many years ago, we developed a mouse model of traumatic stress, which is based on unpredictable maternal separation every day for 3 hours per day, so unpredictably, anytime during the day, combined with unpredictable maternal stress. So the acronym is MSUS. So this is a very extreme manipulation that we do from 1 day after birth until 14 days after birth, so for PND 1 to 14. And we've been phenotyping this model in the past 20 years, and on this slide I will show you the result of these many years of studies on this model. We derived Patry lines by breeding males exposed to wild-type naive females, then taking the offspring, breeding these F1 male whose father has been exposed to wild-type naive females, and then generate multiple offspring. And we've done this experiment 37 times in 37 independent cohorts, and one of these cohorts was bred until F6. So the results of these analyses showed that there are phenotypes which are very consistently transferred from one generation to the next. And here it's a summary of the most significant phenotypes, and risk-taking is one of the behavior which is transmitted until the sixth generation in the Patry line. In the Patry line, we don't know. We know that it's, because we did not do the experiment, we did breed them until the second generation. But in the Patry line, risk-taking, and it makes a lot of sense for humans, because borderline personality disorder, which is a psychiatric disease which is most often associated with childhood trauma, physical violence, sexual violence in early life, patients show a lot of risk-taking behaviors. Metabolism is also significantly altered across multiple generations, altered glucose, insulin, altered weight. The animals are also depressed till the third generation, and then it disappears, this phenotype. Another phenotype that we have unveiled very recently are cardiac dysfunctions. So we worked with a cardiologist, a colleague, who did a cardio MRI on these animals. So it cannot be biased by any influence behavior. You may always say, yeah, maternal care and so on. We track that by doing cross-fostering, for instance, but cardiac dysfunctions, you cannot bias that in a way or another. So we think that this phenotype is really, it's very severe, and it's quite striking that early life trauma induces cardiac dysfunctions. So here I'd just like to quickly show you, just for you to have an idea of how we test risk-taking. We've used the elevated plus maze, which is a task normally used to test anxiety, but we measure, so it's 2 open arms and 2 closed arms, and the animals normally stay in closed arms, and they explore the open arms, but after doing a lot of risk assessment behaviors. So we measure the latency, how long it takes them to venture onto an open arm, and we measure the time they spend in closed and open arms. And what we have seen is that in our animals across generations, they spend more time in the open arms and they enter the open arms more swiftly. So suggestive of risk-taking. the cardiac, that's the results of the cardiac hypertrophy. So we see the weight of the heart is increased, and there is also lung congestion. And functionally, we did, our colleague Francesco Panini did a lot of functional analysis and also staining and histology. And the phenotype is that it's very similar to heart failure in human, where the heart of the wall of the heart is thicker and the tissue is fibrotic, so it does not pump blood properly. So now, is chromatin in brain cells affected by early life stress and may correspond to these phenotypes? So what we did was to generate multiple groups in parallel and then sacrifice animals after they have been exposed for 1 week to the trauma, for 2 weeks to the trauma. Another group, we let them grow adult. We take tissues, and the last group, we let them grow adult, breed them, and generate an offspring. So for each stage, we collect germ cells, so either germ stem cells, spermatogonia cells, or sperm cells, or brain, and we have also collected heart, many other tissues. Now, what we have done is to use ATAC-seq. So ATAC-seq is a method where you use transposase. It's an enzyme which cuts the DNA when it is accessible. So by using by ATAC-seq, you can evaluate which part of the genome are open and which parts of the genome are closed. And then it gives a very good index of chromatin accessibility. And by looking at the sequence, you can also guess what are the regulatory sequences, if there are transcription factor binding sites, for instance. And here we expose our mice to the trauma, we collect tissue, and we prepare genomic DNA, and we run the ATAC-seq. And what we saw immediately is that there were regions which were more open, like you see here. This signal is that what you see dark blue is when chromatin is open. So visually, you already see that there is more dark blue in ELS, which is early life stress. So chromatin, and the fragment is 1 kb plus or minus of these regions. So if you have a fragment of DNA, you identify the region which is open, plus minus 1 kb. And you see that some regions are more open, other regions are closed. And this is adult brain. So brain, many, many months after the end of trauma. So it means that these changes has been maintained in brain cells. And they correspond to intergenic regions, regulatory regions like enhancer, promoters, and distal intergenic. So not in the genes themselves, or very little in the genes themselves, but the regulatory genome. And some of these are regulatory sequences which are important for epigenetic regulators. This seems to be functionally relevant because this is associated with changes in transcription. We don't have a causal relationship directly. For this, we would need to manipulate chromatin accessibility, let's say with CRISPR, and then see whether this has an impact on gene transcription. But on gene transcription, we see that there are genes which are downregulated, others which are upregulated in hippocampus and prefrontal cortex. Some of these genes are specific to hippocampus or prefrontal cortex, and some of the genes are changed in both structures. And again, this includes some epigenetic regulators. Now, an interesting thing, I mentioned enhancers, and we do see that chromatin accessibility is changed in enhancers. Here you have H3K4 monomethyl. It's a histone modification which is present on enhancers. And this is a differential accessibility at the level of enhancers, which is altered in brain tissue, in hippocampus from traumatized animals. And indeed, if we look at the level of expression of enhancer RNA, we see that some enhancers RNA are more abundant and others are less abundant. So, and these are very important transcriptional regulators and genome regulators. So we don't know exactly what they do. We now are using again CRISPR in cell culture and hopefully in vivo to really address the causal relationship between these enhancer RNA and brain functions. Now, are there also chromatin changes in reproductive cells? So This is a very simple diagram of germ cells, male germ cells development from very early on during embryogenesis to adulthood. And the step that is very important to us is the one which covers the window of exposure. And our window of exposure is postnatal day 1 to 14. It's a time when in testes there are almost only germ stem cells, spermatogonia cells. And germ stem cells during that stage and later on, they exist in two types. There is one stem cells which are real stem cells. They renew themselves throughout life, even in adult. And the second type are stem cells which come from the renewing stem cells, but they start to differentiate and will then become spermatocytes and give rise to spermatogenesis. So it's difficult to distinguish them experimentally. So we isolated both of them together by FACS from testes of animals exposed to trauma. This is again the same diagram as before, just for you to remember that we extract them at 1 week after trauma exposure, 2 weeks after trauma exposure, and in adulthood. And we also extract sperm from, in adult, sperm from the exact same male. So we can also compare the data across males. And what we observed, it's known that chromatin accessibility changes, is dynamic during spermatogenesis. And we just confirmed this here. It's in control spermatogonia cells. You see that in adult spermatogonia cells, there are some regions which are more open than in PND15, and vice versa. There are regions which are closed while they are open in PND15. And what we observed in spermatogonia cells from mice which have been exposed to trauma is also an alteration of chromatin accessibility, both at PND 15, PND 8 as well. Here I'm showing PND 15. So already after 2 weeks of trauma, chromatin accessibility changes in many different loci in these cells. And some of these changes, they persist until adulthood. And here it's just a ChIP-seq on the right, ChIP-seq from the literature, which tells us what histone modifications are present normally in the regions where we see this altered chromatin accessibility. So we can infer that these regions where with altered chromatin accessibility, they are kind of poised. They have a little bit of modifications which are normally associated with gene transcription, a little bit of histone modifications which are associated with gene repression. So they are in an intermediate state. They keep memory of prior changes in the epigenome in this region to be ready for the future. Some of these changes, they are maintained in mature sperm. So you imagine that, remember this diagram, you have spermatogonia cells. They go through many, many cycles of mitosis. They go through 2 meioses to give rise to sperm. Despite this, some of these changes in chromatin accessibility, they stay. They are still present in mature sperm. So which says that there are mechanisms which allow to maintain, to keep changes in chromatin structure across development, across mitosis and across meiosis. Now this appears also to be functionally relevant because now if we look at gene transcription in spermatogonia cells during postnatal development and in the adult, we see many changes in gene transcription. Again, it's only correlation between these changes in chromatin accessibility and transcriptional activity, but nevertheless, RNA is changed, is altered. It's not always the same RNA, which are altered across development or in the adult, but there are many different loci. And very importantly is that in sperm, in mature sperm, consistent with the fact that changes in chromatin accessibility were also kept in mature sperm, the transcriptome in mature sperm is affected at all levels, small RNA and long RNA. And if we compare now the RNA in spermatogonia and sperm cells which are similarly altered, we can identify a few targets, which are altered origin spermatogonia and stay altered the same way, the same direction, either decrease or increase in sperm cells. So which is a very important confirmation that there is a link between germ stem cells and sperm cells. 2 minutes, okay. So now I'd like to quickly go through work we did in the past. All the work I just showed is not published, but this one was to prove that sperm RNA is causally related or causally responsible for transmission. So what we did was to extract total RNA from the sperm of traumatized males and inject this total RNA into embryos, control fertilized embryos, and then generate an offspring by surgery after transferring these sperm RNA injected oocytes. And what we observe in the animals which arise from these oocytes or zygotes injected with sperm RNA is that they show phenotypes of trauma, risk-taking and depressive phenotype, and their offspring as well. And that the metabolic phenotypes also reproduce this decrease in increase in glucose and altered glucose insulin metabolism. Yes, I'll skip this, but the conclusions from that is that brain and germ cells can be persistently affected by early life stress via changes in chromatin accessibility or in correlation with changes in chromatin accessibility, and that changes are likely functional and are reflected in the transcriptome, because depending on its level of opening, chromatin will be accessible to transcription factors, transcriptional regulators, and transcribed or not, including enhancer RNA. And that sperm RNA is causally linked to the transmission of symptoms of exposure from father to offspring. Just in between brackets, recently I told you we generated the matriline, so transmission through mothers. We repeated all these experiments in oocytes. We could see that the transcriptome of oocytes and chromatin accessibility in oocytes is altered. And also if we inject RNA from oocytes from females who have been traumatized into control zygote, we can also reproduce symptoms. So it seems to be a general phenomenon that gamete RNA can be modified by exposure and keep traces of the exposure for transmission in the following generation. And the causality between changes in spermatogonia cells and sperm cells still need to be demonstrated. Something I will not have time to talk to you about, but is that blood, we believe that blood is or blood components are the factors which connect what captures the brain, what the brain captures, stress in this case, and germ cells. And because we have a study that we published recently where we could show that if we inject serum from traumatized males IV into control males, their offspring show symptoms of a trauma. And we have identified some of these components. Now, quickly, perspective for humans is an important gain in knowledge in some mechanism of memory of prior experience and on how the environment, in particular involving trauma, can influence the organism. Candidate for diagnostics, and we are also working on that in humans who have been traumatized and drawing RNA signature from blood. EVs in extracellular vesicles, because we know that EV signatures are very specific to trauma, and with potential for therapeutics and perspective for evolution, which I don't really have time to go through. And this is my lab. And the most important women who have developed the model, phenotyped this model, and did amazing work. And I can't resist to share my very personal contribution to epigenetic inheritance, and with my own matriline with 4 generations. And a little touch about art. Because during the confinement, this artist, Sandrine Dognaud-Renaud, who is a French artist, she heard a public talk that I gave. She heard it on YouTube, and she was very inspired also by her own past, and she built this beautiful sculpture. So it's there are heads. You may not see that, heads of different age that she disposed in as a double helix. So the sculpture, she then wrote me an email, say, look, I mean, you may be interested in seeing this sculpture that I made inspired by your work, and I was really amazed by the beauty of this sculpture. It was exposed in 2021 in the Grand Palais. She even won a prize. And I went to this exhibition, actually, together with Michael Skinner, the Mr. Epigenetic Inheritance, who was teaching in Paris at the time. And if you are interested, she made a movie here. You have, if you go on her website, it's a beautiful movie where you see the sculpture in this destroyed Parisian apartment, and with the text, and it's very nice. I bought the sculpture, and it's now in my lab, actually. Last thing, we are organizing an epigenetic inheritance symposium this August, in 2 months from now, and you are all welcome to register if you want. It's in person presence, but in confidence, I think I will open online participation maybe a few weeks before the symposium. So if you are interested, you cannot go, you can still look at the website in about a month and register. Yeah, thank you. Sorry for being over time.
So yeah, that was really cool. I'm just wondering if you're in your work in the blood component of this may be a reaching question, sort of gives you a sense for kind of where the specificity might come from. Because it's not like a global change, it's not like everything, it's like certain things are changing, certain genes are being upregulated or downregulated. So that implies that there's evolved some kind of very specific response that may have evolved to kind of ensure that offspring are ready for traumatic environments or something like that. So I'm just curious if you know anything about where that specificity might derive from.
I'm not sure if we can really talk about specificity, because if we look in any organ in these animals, there are genes which are changed. The immune system is changed, hematopoietic stem cells are changed. So, and when we look at the blood composition, lipids are altered, proteins and neuropeptides. So I understand that the phenotype is, I mean, there is still some specificity, but I can't really tell, because they may, the effects are different on different organs. But I mean, that's all we know now about the specificity.